So right here again is our replication fork. Okay. What would be the enzyme that is up ahead and helping to prevent overwinding? This enzyme up front. Topoisomerase. Okay. Looks like right through here I have a protein, another enzyme if you will, that is helping to prevent winding right at the replication fork. What's that called? SSBP, single strand binding protein. Okay. And then we can notice a couple of important things. First off, I recognize this enzyme because do you see how it's adding those first couple of bases that are RNA? What's the name of this enzyme right there that's adding only RNA to the growing strand? Or I'm sorry, not to a growing strand, but a beginning strand. Yeah, RNA primase. And it adds RNA nucleotides originally. I think we might want to label our sides real quick. So which side appears to be leading on this replication fork? Would you say the top strand that's adding to the replication fork, or is it the bottom strand that's leading? I'd say that this one right here is the leading strand. Remember, a strand is leading if it can work in the 5' to 3' direction perfectly, correct? Which means that this top dark black strand has got to be what prime? 3'. And this strand right here is 5'. So that means that this bottom original strand is 3' to 5', which would make each of these beginning pieces a 5' to a 3', a 5' to a 3'. 3' over there, that's 5. Cool? We get how we can see that in our minds, right? Is there any question about that? For sure on the exam, one of your essays, I promise you, will be to draw this. Or to take a picture like this and make it happen. You set? Okay. You'll notice that that's not the only place where you have the RNA primer. You've got RNA primer that's right here. That means that RNA primase was once doing a job right there. And you'll notice 3 times over that you have DNA polymerase. Why don't you take a second real quick and draw in the 3 spots where you believe DNA polymerase is. I see 3 DNA polymerases on this picture. I think we got one that's right here. What does DNA polymerase do? Adds DNA, new nucleotides, right? It actually polymerizes the reaction of adding nucleotides to the DNA. So in these 3 pieces, we have the DNA polymerase. Right? We need to talk about one more enzyme, actually 2 more. And this is going to sound like just a tad awkward, but we'll do it anyway. There's this enzyme that's right here that we haven't accounted for. And I would say that this enzyme right here is called DNA ligase, because it appears to be gluing the strands together. In fact, that's what connects all of your little fragments together. Remember, on your lagging strand side, which again, these pieces right here would represent the lagging strands. They're made of fragments. Do you know what those fragments are called? Okazaki fragments. Right, Connor? Okazaki fragments. So the lagging strand, the little pieces on the lagging strand, little pieces that are made, which you're seeing right here, this little fragment, Okazaki fragments. Good? Okay. And so ligase is going to be the enzyme that will glue everything together. Done with this picture? So we might want to jot down that the lagging strand is made of units of nucleotides. What is it? Okay, so the lagging strand is made of units, or we could say fragments, of nucleotides called Okazaki fragments. So these are the little units. And if you think about it, they're formed because you got to go towards the replication fork. But with that being said, you still have to, like, you can only go in the 5 to 3 direction. So you would have these little fragment pieces on one side. As we just had mentioned, DNA ligase will connect these pieces. Also, I think it's important that we not forget that RNA nucleotides were used along the way to prime. And so after a while, it could be expected that they would have to be replaced. Another form of DNA polymerase, which we don't have to know the different forms, so we'll just go with the fact that it's just DNA polymerase in general. Will replace the RNA nucleotides. Okay, so I think the other thing to remember is that there has to be a system that is helping to fix or make sure that we're doing everything correct on this. So I think we could also mention that DNA polymerase, as though it didn't have enough forms and enough functions, it also proofreads and verifies that the correct base is added. Then it will actually, it will usually replace them. If it's improper, if it's the improper nucleotide. And I shouldn't have said correct base. It's the correct nucleotide with the correct base. If the improper nucleotide is there, DNA polymerase will replace it. So remember that when we're looking at this little piece right here, and I'll zoom in real quick, or I'll just show you. DNA polymerase is about to add a new nucleotide at this position where my pin is at. If this original parent strand, it's the complementary of it, if you will, is a C, then which nucleotide should be added at this spot right there? A G. And if this next one has an A on the parent strand, then this should be a T that's added initially, or the T that's added right there, right? And it's not, you're not adding bases, you're adding nucleotides. It's just that nucleotides contain bases as one of the 3 parts. Correct? Now, speaking of incorrect stuff, there is a method of ruining your DNA. Like, you can actually hurt your DNA. And so we could agree that DNA damage could be caused by can be as 2 words, obviously.